Okay. Morning, everybody. Yeah, you guys really, I really need to give you guys a extra points, right? Coming today. All right. Um, yeah. Some of you guys asking about uh, the paper for due Friday, and whether I think you guys need to submit a full research paper. That means 6 pages, right? Just wanted to clarify. Uh, and okay, so today we're going to talk about the last part of the lipid metabolism, essential fatty acids. So essential fatty acids, the definition means that the type of fatty acids that our body is not able to synthesize. That means we need to acquire this type of fatty acids from the diet, right? So this is the definition. So it means that we cannot synthesize in the body, but need to get these fatty acids from the diet. And essential fatty acids usually are the polyunsaturated fatty acids, right? Because the human bodies lacks of saturated delta 12 and delta 15, right? But we do have some desaturated. Remember for the formation of double bonds, and we require these desaturates, right? And desaturates, and we have a different type of desaturates depend on the position of double bond. For example, we do have a delta 5 or delta 6 desaturates. That means these desaturates can catalyze the formation of double bonds at delta 6 or delta 5 positions, but we do not have delta 12 and delta 15 desaturates, so that we cannot synthesize some of the polyunsaturated fatty acids in the body, so essential fatty acids, right? There are two classes of essential fatty acids, omega-6 and omega-3. And omega-6 essential fatty acids is linoleic acid. Linoleic acid, which has 18 carbons and 2 double bonds, is the omega-6 essential fatty acids, right? So, and this linoleic acid mostly present in the diet, very abundant in the diet. So we are able to acquire the linoleic acid from the diet. And there's also other family members, omega-6 unsaturated fatty acids. These are arachidonic acids and arginic acid. And these two polyunsaturated fatty acids are able to synthesize from linoleic acid in the body, right? So I just wanted to point out that only linoleic acid is an essential fatty acid, right? Arachidonic acid or arginic acid are not essential fatty acids because we are able to synthesize in the body from linoleic acid. And similar for the omega-3 essential fatty acids, alpha-linolenic acid, 18 carbons and 3 double bonds, and this alpha-linolenic acid is essential fatty acids, right? And the other two, EPA and DHA, are the omega-3 family members of polyunsaturated fatty acids, but they are not essential fatty acids. They are able to be synthesized from alpha-linoleic acid in the body, right? So we only have 2 essential fatty acids, one from each category, omega-3 and omega-6, right? Alpha-linoleic acid or alpha and linoleic acid and alpha-linolenic acid, right? Essential fatty acids. Is that clear? Right? And essential fatty acids, if we compare to the non-essential fatty acids, they have a different function. We need both, and non-essential fatty acids like saturated fatty acids or monounsaturated fatty acids. This fatty acid usually we use as energy fuels, right? Via the beta oxidation, and these fatty acids usually stored in the form of triglyceride as energy, right, in the adipose tissue. And essential fatty acids usually are not metabolized for energy use. This is not the main purpose of essential fatty acid for energy. Right? It's not main purpose, but it's very important component of cell membranes, and it also involves some important biological processes. Right? I just wanted you guys to review the structure of fatty acids to get better understanding the structure of the polyunsaturated fatty acids. For example, here we give the saturated fatty acids as an example. Usually we have fatty acids as a carboxyl end and a methyl end, and also hydrocarbon chain, right? And also numbering system, the omega numbering system. Usually the first carbon at the methyl end we named as omega 1, right? Remember this numbering system. And so omega basically refers to the first double bond position starts from the methyl end. For example, omega-3 fatty acids usually have a first double bond at the omega-3 carbon positions. And omega-6 unsaturated fatty acids has a first double bond at the carbon 6 omega-6 position, right? So this is how omega fatty acids, 3 or 6 fatty acids are named. So depend on the first double bond, the position of the first double bond, right? And here just give you kind of structure of the just two omega family members. The omega-6 fatty acids, again, the linoleic acid is the essential fatty acids, and arachidonic acid is omega-6 fatty acid family, but it's not essential fatty acid, can be synthesized from linoleic acid, right? So linoleic acid has two double bonds at 9 and 12. They are also a different number system, so delta number system usually used to name the double bond position, and this double bond position is start from the carboxyl group, right? Carboxyl end. For example, this delta 9 means that the double bond at the position, the carbon 9 from the carboxyl group. Remember, we have also the numbering 1, 2, 3 numbering system, right? So, it's kind of I don't know why they use so many different numbering systems, but just and so the two double bonds in the linoleic acid at delta 9 and delta 12 position, right? Carbon position. And arachidonic acid has a 20 carbons and 4 double bonds at delta 5, 8, 11, and 14 carbon position, right? And omega-3 fatty acids, alpha-linolenic acid, is essential fatty acids, again has a 3 double bonds, 18 carbons, and EPA is the members of omega-3 fatty acid family, but it is not essential fatty acids. Again, it can be synthesized from alpha-linolenic acid, right? So omega-6 polyunsaturated fatty acid family, we know have a major 3, right? linoleic acid, which is essential fatty acids, and arachidonic acid and arginic acid. And these three polyunsaturated fatty acids are the omega-6 families, right, members. So linoleic acid, the main sources of linoleic acid, and we know that we have a lot of linoleic acid in the diet. The basic is from corn oil, sunflower oil, seed oil, and safflower oil, and most abundant. And so linoleic acid is most abundant polyunsaturated fatty acids in the diet, right? And arachidonic acid can be synthesized from dietary linoleic acid, and arachidonic acid is main substrate for the synthesis of eicosanoid biomediators, for example, prostaglandins and leukotrienes, right? So arachidonic acid is very important. The omega-6 family members, it can be used to synthesize a lot of different type of the eicosanoid biomediators. For example, again, the prostaglandins and leukotrienes, right? And arginic acid usually can be converted from arachidonic acid by just the elongation reaction, because it just have two more carbons, right, than the arachidonic acid. And it can also be converted back to arachidonic acid, right? So I think that the arachidonic acid is more important than the arginic acid. These two fatty acids can be interconverted. And omega-3 polyunsaturated fatty acids the family has the three members: the alpha-linoleic acid, which is essential fatty acids, right, and EPA and DHA. And alpha-linoleic acid is also can be get from the diet. For example, soybean oil, canola oil, and vegetation. And EPA and DHA mostly the most abundant sources of EPA, DHA actually are the marine animals like fish oil, right? So metabolism of fatty acids, essential fatty acids. First of all, our body uses the dietary linoleic acid or alpha-linolenic acid, which two essential fatty acids, to synthesize all other forms of omega-3 and omega-6 fatty acids, right? I described a lot. So this is the first, how essential fatty acids being metabolized. So we get the essential fatty acids from the diet and use these two essential fatty acids to synthesize the other type of members of omega-3 and omega-6 fatty acids, right? And so for the synthesis of omega-3 and 6 fatty acids, there are several reactions required, including like elongation, desaturating, and Ricci conversion, because of the other members of omega-3 and omega-6 fatty acids have a longer carbon chain and also more double bonds, right? So that it requires elongation to extend the carbon chain, and also desaturation to add more double bonds to the molecules. And these two omega-3 and 6 family members, they cannot be interconverted, right? They kind of separate, the category or types of fatty acids. They cannot be interconverted. But within the same family members of fatty acids, they can be interconverted. For example, we already talked about arachidonic acid, arginic acid, they can be interconverted, right? And this slide just show you kind of overall pictures of how different categories or families of omega-6 and 3 fatty acids being synthesized. Let's look at the omega-6 first. So linoleic acid serves as the substrate, right? It is essential fatty acids, and through the it can be used to synthesize arachidonic acid. So if you compare the carbon chain and the double bond numbers, that you can see the arachidonic acid has a 20 and linoleic acid has a 18 carbon bond, right? So we need to extend at least, to add at least 2 carbons to the arachidonic acid, and to make to add linoleic acid to make arachidonic acid, and also we need to add to create 2 double bonds, right? And to make arachidonic acid. So you can see that first of all we need to desaturate. So delta 6, delta 5 that our body has, right? The desaturates, and undergoing the 2 desaturation steps to adding 2 double bonds to the linoleic acid, and then elongates to catalyze the extension of the carbon to add 2 carbons to the linoleic acid and to form arachidonic acid. And you also go to the further elongation and desaturation to add more carbons and 1 more double bonds, and to make the 24 the fatty acids, which has 24 carbons and 5 double bonds. And arginic acid, this arginic acid, right? Arginic acid usually has 22 carbons and 5 double bonds, and this can be literally converted back to and this 24 carbon and 5 double bond fatty acids can be converted back to the 22 double bonds, right? And 22 carbons and 5 double bonds, right? Which is arginic acid. And then omega-3 fatty acids, family members can be synthesized from alpha-linoleic acid, which is essential fatty acids, 18 carbons, 3 double bonds, and to make DHA, and DHA has 22 carbon and 6 double bonds, right? And to undergo, the 3 desaturating and 3 extension steps, and to make, the longer to make DHA, which has a longer carbons and more double bonds, right? So you don't have to, I guess, remember all the details, but just get a sense, the other family members of the fatty acids can be synthesized from linoleic acid and through the desaturation and elongation steps, right? And this just gives you a little bit detailed, to help understand the process of the synthesis of the omega-6 saturated fatty acids from the linoleic acid, right? So, and first reaction is the substrate is a linoleal-CoA, right? And the first reaction is desaturation, so delta, I think delta 4, right? Can't see. So you can see, sorry, delta 6, right? It's too small, I cannot see. It's 4 or 6? 6, right? Okay, yeah. I think it's 6, right? See, yeah. So the first reaction is a desaturations. So delta 6 desaturates to catalyze the addition of the double bonds at the carbon 6 positions, right? So you can see. So then you, this molecule has 3 double bonds. And then the next step is the elongation. You add carbons, 2 carbons, and the sources of a carbon donors is marino-CoA, if you remember the fatty acid synthesis, right? Marino-CoA is serve as the carbon donors. So you extend the 2 carbons between initial carbon 6 and the carbon 1 positions, right? So then you can see the switch, the position switch of the double bonds from 6 to 8 and 9 to 11 and 12 to 14, right? Because the 2 carbons is added between this first the double bond, first double bond and the carboxyl group. And then additional desaturations, the delta 5, right, desaturates and to catalyze the formation of double bond at the carbon 5 position. So eventually, you undergo 2 desaturating reaction and 1 extension, elongation reactions, and to make the arachidonic acid, right? And again, this whole process requires the marino-CoA as the carbon donors, and need a delta 5 or delta 6 desaturates to catalyze the formation of double bonds and also elongates for the extension of the carbon chain. And it also requires the NADH and NADPH, right? Any questions? Okay, so some more information about the omega-6 polyunsaturated fatty acids. And we know that arachidonic acid, the essential fatty acid, linoleic acid, usually have a little bit biological effect. So mostly it's just used for synthesis of the other family members of fatty acids. For example, here the arachidonic acid, because arachidonic acid is very important, the fatty acids that involve a lot of biological activities, right? So here, arachidonic acid can be synthesized from linoleic acid, and arachidonic acid is very important substrate for the synthesis of eicosanoids, including the prostaglandins and thromboxane and leukotriene and the other mediators. And these eicosanoids are the very important bioactive molecules that involved in the biologic process, particularly the inflammation. So that's how omega-6 fatty acids that regulates the biological function. And specifically, and here I just want to mention again that the omega-6 polyunsaturated fatty acid function, the linoleic acid, and in addition to its function as the precursors or the substrate for the synthesis of arachidonic acid, and it's also itself can be as the component of a single lipids, and also serve as the skin water barrier. And arachidonic acid, as I mentioned, is the primary substrate of the eicosanoid biosynthesis, and it's also a major component of phospholipids, which is a very important signaling transduction, involved in signaling transduction. And also omega-6 fatty acids and some eicosanoids can be served as the ligand for the transcription factors. PPAR gamma is a key transcription factor that regulates lipid metabolism, right? And so by affecting the PPAR gamma activities, so this eicosanoids can regulate the lipid metabolism. So arachidonic acid, and this slide just show you how the process, a little bit detailed process, how arachidonic acid is used for synthesis of eicosanoids. And there are actually 3 enzymes that involve this process. They're called cyclooxygenase, lipoxygenase, and cytochrome P450. And these 3 enzymes are very important that mediates the production of the eicosanoid from arachidonic acid. Right? This eicosanoid including the prostaglandins, the leukotrienes, and these are the very important mediators of a local pro-inflammatory response, right, reactions. So, this again, this bioactive compound that involved in the inflammation, inflammation in the local inflammation, inflammation in the tissues, organs, right? Now, how about the omega-3 polyunsaturated fatty acid? So omega-3 alpha-linoleic acid itself doesn't seem to have a unique functional effect, but again, it is very important precursor, right, substrate for synthesis of EPA and DHA, the other family members of omega-3 fatty acids. And similarly, EPA and DHA can also be used for synthesis of bioactive compounds, and also use the same pathway, same set of enzyme, right? Cyclooxygenase, lipoxygenase, and cytochrome P450, and use the same set of enzyme to synthesize the different bioactive compounds. They are called resolvins and protectins. And resolving protectins are a number of the molecules, bioactive molecules that are very important, that involve anti-inflammatory response, right? So you can see that omega-3 and omega-6 family members, that they are involved in different, biosynthesis of different sets of the bioactive molecules, right? And involve, the omega-3, 6 fatty acids derived the bioactive compounds or molecules that involve inflammation, right? And then the omega-3 fatty acids derived the bioactive compound that involve anti-inflammation. So they have a different effect, functional, biological function. So that's why we heard a lot, right? The omega-3 fatty acids are considered good fatty acids, EPA and DHA, because this is the main reason, because they involve a different, the biological process. But these 2 family members, they have a functional relationship. Remember, both omega-3 and 6 fatty acids, they use the same set of enzyme, right, to synthesize the different type of eicosanoid bioactive compounds. So these 2 type of fatty acids, they can compete for the metabolized enzyme, right? They compete for these 3 major enzymes for synthesis of omega-3 eicosanoid or omega-6 eicosanoid. For example, if we have a lot of EPA omega-3 fatty acids available, then the more the set of enzymes are used for the synthesis of omega-3 eicosanoid, right? So we have a more, which is good, or anti-inflammatory eicosanoid being synthesized, then we can reduce the production of the bad or pro-inflammatory omega-6 eicosanoid production, right? So this sort of kind of mechanism, how EPA have an anti-inflammatory effect, right? So because we compete for the same set of enzyme to have a good, to have a more the since production to produce more good eicosanoid and reduce the production of a bad eicosanoids, right? So EPA, the omega-3 fatty acid function, as I mentioned, it's a smaller molecules and less bioactive compared to the the omega-6, but it's very important. As I mentioned, you compete for the metabolized enzyme, right, to inhibit omega-6 eicosanoid production, and because of eicosanoid, omega-3 eicosanoid are the pro-inflammatory, right? Sorry, the anti-inflammatory. So EPA has the anti-inflammatory functions. It can increase. Some lot of study also showing that EPA, if you consume a lot of EPA, that can promote fatty acid oxidation and to reduce the triglyceride levels in the plasma. It can also, inhibit the hepatic triglyceride synthesis and reduce the fatty liver disease. And DHA function. So DHA usually present in the phospholipids of cell membrane. So it's pretty enriched in the retinol and brain. So it's very important for the visual and cognitive actions, and can increase the plasma logins. And this, because this is the class of a membrane phospholipids, they function, the membrane vesicle fusion, intracellular signal transduction, and protect against oxidative stress. So increase these molecules, it should be good for the health, right? So that's just in general. I mean, we don't want to go to the details. So okay, so I think that this probably is the last slide of the this essential fatty acids. And essential fatty acids is important because they require for the synthesis of arachidonic acid and its derivative, right, the eicosanoid. And this eicosanoid molecules are very important and can regulate a set of transcription factors, and that control the genes that involve lipid and glucose metabolism. For example, SIBP, you guys notice that, right? This is an important transcription factor, remember, control the HMG-CoA reductase for the cholesterol synthesis. So the eicosanoid can regulate these SIBP activities and can regulate cholesterol synthesis. And PPAR gamma, PPAR, a set of transcription factors that can regulate the lipid lipogenesis and also the some other the sets of enzymes that involve the lipid metabolism. So I guess this probably is the last slide. Yes. So just go through the summary. Again, the linoleic acid or alpha-linoleic acid are the essential fatty acids and must be provided by the diet because we cannot synthesize this two essential fatty acids because we lack of the desaturates, right? Remember which desaturates that we don't have? Delta 12 and 15, right? And which set of desaturates that we have? We do have 5 and 6. So try to remember 5, 6 we desaturates that we have, and 12 and 15 desaturates that we don't have. And human bodies, cannot compete the synthesis of LA and alpha LA. And omega-6 family polyunsaturated fatty acid can be synthesized from linoleic acid, right, through the two important reactions, desaturating and elongation. And the main component of the omega-6 families is the alpha-linoleic acid, right? And alpha-linoleic, sorry, I don't know why, arachidonic acid, right? So arachidonic acid is the substrate of the eicosanoid or the pro-inflammatory eicosanoid synthesis, right? And which is a very important bioactive molecule that involves the pro-inflammatory response, so which is considered not good, right? Not is a bad eicosanoid. And omega-3 families can be synthesized from alpha-linoleic acid, again through the same reactions of desaturating and the elongation. And EPA and DHA are the two major forms of alpha-3 fatty acids family. And the eicosanoid synthesized from the EPA has an anti-inflammatory function, right? So these two alpha-3 and omega-3 and omega-6 family members can be compete for the set of enzyme that for the biosynthesis of the eicosanoid. So the reason that EPA has an anti-inflammatory function is because it is a substrate of the anti-inflammatory eicosanoid synthesis, right? And it also can compete with the omega-6 family members and to reduce the pro-inflammatory eicosanoid synthesis. All right, so I think that's all for today. Do you guys have any questions specific for the, for the exam? Or are you guys ready for the questions? Or okay. I don't think we will have any structure in the exam. You probably don't need to remember the structure, but I think the structure sometimes help you understand the stuff. So, yeah. Yes. Oh, that's a good question. No, I don't think. No, not specific. Any other questions, or do we kind of? Yeah.
Quick clarification question. You had said that the adrenic acid was 22 carbons with 4 double bonds on
Yeah.
one side, and then you said it was 5 double bonds on the other one.
Really?
So just check.
Oh. In which slide?
One,
Oh,
is
this.
it 4 or is it 5?
Let me see, what is it? Other sets 4? Oh, okay. Ah, that's a mistake. Um, hmm. I think should be 5, right? I will double check.
Okay.
Sorry about this mistake. uh, any other questions? No? I guess you guys probably will have a more question next Tuesday, right? Okay. So yeah, just use the rest of time for your, working on your papers, right? All right.